Hello, let's talk about extended shelf life of semen dose, Stallion semen dose. It is a very valuable tool and we will understand how it's important, how it was possible, how it can be used in our daily practice, all the things involved to reach this level, and also how it's not fair with some stallions to say that they are bad coolers, for example. Another thing is, it's not new. It has been used in different species. And now it's our turn to understand this technique and to explore it as best as you can to make our life in the daily equine reproduction practice easier. Let's go. Let's talk about extended shelf life on Stallion chew doses as a very valuable tool. I have been visiting different farms, different countries, and different stallions as well, of different breeds. And in general, and it will not be different here in the Middle East, that also the Arabian stallions are labeled as bad freezers or bad coolers. Something that is absolutely unfair. All the breeds that I had the opportunity to work with, you have stallions that are good and are bad. The semen are good, the semen are bad. The most important thing is to keep in our mind that all of them, they are different. And we cannot consider them all as the same. We cannot use the same standardized protocol for all of them, ignoring their individual preferences. This is one thing to keep in mind to avoid labeling a stallion, a good one that could produce very good semen doses, as a bad one. As I said, it is not a new thing, this extended semen dose. In porcine boar industry and dogs industry, in both species, they are using that technique since a long time, even having a very different kind of semen. Dog semen is well known as a very tough one, and boar semen is well known as a very weak one. Dog semen is can survive at very deep temperatures. So it's not that sensitive to this temperature drop. In boar semen, it's not like that. The temperature has to be a little bit warmer. However, in both, the technology adjusted the protocol and the extenders to reach the demand. In both situations, to have semen with a longer survival was important, as for us in equine industry. And from now on, we can do it as well, and let's talk about it. Let's start with the difference among chilled doses and frozen doses. Chilled doses are, for sure, cheaper to storage, easy to transport, easy to do the insemination. They have a long intrauterine lifespan, 48 hours, even more, and this is the tricky part, a short shelf life. What about the frozen semen? It's more expensive, it requires liquid nitrogen container. You can use it only for deep horn insemination if you want to reach good numbers. You have to know the precise moment of the ovulation. It has a very short lifespan into the uterus, so you have to scan the mare more often. The main advantage, it have a long shelf life, and the sperm can be there storage for many years, even decades. So they are different. I'm not saying here that one is better than the other one. However, right now we are almost 100% focused in the frozen semen if you want to keep the semen more than, let's say, 3-4 days. Can be more if you improve the shelf life of the chilled semen. And this is what we're going to talk along this conversation. Many things has to be considered when we are talking about the sperm longevity. The sperm morphology, concentration, nutrition, oxidation, osmolarity, bacterial load, seminal plasma, the sperm membrane itself, the storage temperature, and the cooling curve. All these things are working together, and each one of them are adding value to the semen quality, improving or not. For example, morphology. If you have a sperm with a bad morphology, for sure you also have some other damage, and it will not be good for the environment of the dose, and you have to remove them if possible. Concentration. Concentration is another thing that you have to consider. A high concentration means that maybe you not have enough nutrition, or you're going to have a lot of production of reactive oxygen species that will be harmful for the environment and for the sperm that are there. Oxidation is absolutely related with all of this. Nutrition, because of the metabolism, produce ROS, and the concentration as well. Oxidation is part of the process, has to be controlled. Osmolarity, the same. Bacterial load. Another thing, the bacteria there in the sample is not good. It will be harmful to the other sperm, to the environment, and it will also produce a lot of reactive oxygen species. It is something that has to be considered. What about the seminal plasma? In the nature, the seminal plasma is not with the semen for a long time. But when you are talking about the semen doses in general, if you don't process it, it will be there for a long period, and it will not be good for the sperm as well, and has to be removed or considered. Sperm membrane, storage, the temperature, and the cooling curve. Well, it's well known that you have individual species characteristics on the sperm composition. Different amount of cholesterol, lipids. It is all involved in the thermal resistance of the sperm. It's well known. And you have to address the difference among species, but also for each individual characteristic when you are talking about different stallions. If they can survive better at 5 degrees, at 15 degrees, 17 degrees, it all has to be considered as well. The cooling curve is the same way. If you drop the temperature too fast, some stallions will not survive, and you have to figure it out. Eventually you have to go slowly, and then the sperm will have a better chance to survive. And the temperature as well, the storage temperature, you have to consider that. You're gonna keep, because the temperature is absolutely related with metabolism. When you drop the temperature to liquid nitrogen to less, to minus 120 degrees or even lower than that, the temperature, well, the metabolism is very low, almost zero, let's say that. So all the production of ROS is stopped. When you go to 15 degrees, it's less tough for the membrane, but the metabolism is quite higher than the 5 degrees. So you have to consider and to have a good balance. Stallion, protocol, extender, to figure it out the best approach to keep those semen, those sperm cells alive for a longer time. When you compare the way that the freezing, the sperm freezing and being done nowadays, many steps are being ignored. One thing that I just mentioned was the cooling curve. And pay attention how the freezing process is being done in the field. Instead of having controlled cooling curve using a freezing machine to be sure that you not be tough for the sperm cell, in general, it is done with a normal fridge or a nitrogen vapor. That is not that controlled, and some stallions maybe they will be suffering and will be considered as a bad freezer or bad coolers as well. It must be considered. So far, right now, when you are talking about the cooling curve, it's been hard. But what about the temperature storage? It has been difficult as well because so far we were not equipped for using, or we're gonna use a container for liquid nitrogen that is frozen semen, or just neopore box that are able to keep the temperature, let's say 24, 48 hours maximum. Now, to be able to keep the sperm for a longer time, we need to add another equipment. But it is good. We are not talking about improving the freezing and the cost of it using a very expensive machines. We are talking about new and cheap things that will be able to keep the sperm cell for more time. So we are talking about normal refrigerators that are quite easy to find and quite easy to use and to control the temperature at 5, 15, 17, taking care of the sperm to not be suffering with the cold shock, for example. There are basic things that has to be considered when we are talking about semen processing. Doesn't matter if we're going to talk about freezing or if we're going to talk about cooling. First of all, hygiene. Everything starts with washing the penis to remove all the problems, all the bacteria, not all, I'll say, but to reduce the bacterial load. That is a very important thing. Another thing is to remove the gel. You have to remove it in the nature it will not be in contact. Use it by a filtering. So, doing both things, you reduce the bacterial load. So you're going to reduce the contamination. However, another thing has to be considered as well. It's not only about the semen, the collection that should be done, the semen collection that should be done in one jump only just to, let's say, to remove or to reduce the bacterial contamination. Not only that, other things as tubes, as gloves, as the laboratory bench, all of these has to be clean as possible to reduce this contamination. It's a very important part when we are talking about semen processing. Doesn't matter the destination of the semen that was just collected. In general, semen extenders have a basic formula, and how to use it is being done in the quite of standardized protocols. Let's address quickly each one of them and find out some points that might interfere in the results. The dilution, most of the times, are being based in volume. Volume of the ejaculate and volume of the extender used. It's not precise. To be better, it should be done based on the concentration. It should be done with using the concentration to be sure that you have a controlled environment. In general, the extenders, they have sugar, different kinds of, different combination, milk or part of it, egg yolk or other things. Also, this egg yolk and the raw extender can be processed in different ways. And another point that's advantage things, but people are not doing that much in the protocols, but to the addition of cholesterol. Cholesterol in the formula of the extender is something very important because it's well known that the cholesterol provide to the sperm cell, to the sperm membrane, higher resistance to the lower temperatures. But this cholesterol addition is quite complicated. It's not being done in all the formulas. Something that you have to address, you have to keep in mind if you want to improve the sperm resistance to lower temperatures. It's not only about the extender itself, it's about the semen processing. Well, seminal plasma is something that is there in the ejaculate, but can be removed. It can be removed by centrifugation. This centrifugation can be done normal centrifugation or the centrifugation, protected centrifugation, adding a cushion fluid. In these, you have a higher sperm retrieval with less sperm damage because of this cushion that you add in the tube. Antibiotics and antioxidants. Well, the bacteria are everywhere. The addition of antibiotics in the formula is very important. So you have to control the contamination, but you have to add as well antibiotics. We know that there are some issues related to the bacterial resistance, but so far it's quite hard to work without antibiotics in the formula. And the newest thing that I like to say that's a game changer is the addition of antioxidants in the extenders formula. It made a huge difference because, as we already mentioned, and we'll talk more along this presentation, the production of reactive oxygen species is something that occurs normally in the sperm metabolism. And it's very important to control it, to control this oxidation by the addition of antioxidants. Another thing that can be done as well is the centrifugation, but not just the centrifugation and removal of dead sperm and also the sperms that are with some problems in the morphology. As we already addressed, they are not good for the semen dose environment. And we have been working quite a while in the last years in the semen colloid centrifugation. And we have been published many things proving that it works. Most recently, we are working also with the microfluidic sperm selection that also does the same as the colloid centrifugation, however, without needing the centrifugation itself. What the difference of them? The colloid centrifugation, you add the semen sample in the top of the colloid, and after the centrifugation, you collect the good sperm in the bottom of the tube. The microfluid centrifugation, you add the semen dose below this membrane, you add the extender in the top of this membrane, and after 30 minutes leaving it in the warm temperature, the good sperm will cross the membrane and will pass through it and will be collected after that. More than that as well, other advantage you have when you use this kind of techniques. You remove you reduce bad sperm, dead sperm, but also seminal plasma, bacteria, urine, and many other contaminations that will interfere in the future in the quality of this semen dose that you want to process. Recently, a new extender considering most of those points that we already addressed was launched. The name of this extender was ONE. It was produced or developed in the University of Newcastle, that's the name, and it was published a few years ago by Zamira Gibb and her group with Dr. Ike as well, showing that it was the first extender in the world able to keep the semen dose, the Stallion semen dose, up to 7 days. And for that, it was considered many of the aspects that we spoke about: oxidation, temperature, concentration, purification or selection of the good sperms. All of these were considered in this first publication, considering all these weak points that we mentioned before. This is the comparison of the dose after 7 days. This is the initial progressive motility at room temperature, so it's quite warm, and at 17 degrees, showing that at 17 degrees it was good, barely the same as the initial motility and even better than the room temperature. The same, you had a better progressive motility with sperm selected with a single layer centrifugation, and it was good as well. And also in this comparison of the capacitation and also the progressive motility of the one extender in comparison with the milk-based extender, showing that pre-capacitation, post-capacitation, the motility was higher. So in this conclusion, we can notice that it was possible for the first time to keep this sperm with a good motility and also good fertility after 7 days. The Stallion ejaculation have the sperm cells and also the seminal plasma. Going deeper in the understanding of seminal plasma, it's made with many compounds, many substances. You have a lot of proteins, testosterone, and many things are there. And about the effects, the functions of it, it's a puzzle of many things. So the seminal plasma is involved in capacitation, in the immune response, and many other things. So the sperm is dependent of the seminal plasma. It's true. However, as I mentioned before, the seminal plasma is not developed in the nature to be with the sperm all the time. And we have to understand that how important is this semen, this sperm, the seminal plasma removal. Let's figure it out, the effect of the seminal plasma in the store shelf life. In this study, comparing 0% of seminal plasma and 20% of seminal plasma, is easier to identify a higher total motility, progressive motility, and other kinematics parameters of the sperm. So, no seminal plasma removed by centrifugation is better than 20%. Also, comparing this one, just the dilution, not the removal itself, just the dilution, and all these data after 48 hours at 5 degrees, we can see that when we compare the dilution 1 plus 4, 1 part of semen, 4 parts of extender, or 1 plus 9, we can also notice that you have a better motility when you have a higher dilution. So, what it means, higher concentration of seminal plasma is not good if you want to keep the semen for a longer time. So it has to be removed or properly diluted. Let's see another example in this slide. This is the comparison of 20%, 50%, 80% in two situations. After 24 hours, after 48 hours, considering the sperm membrane integrity and also the sperm motility. It's easy to understand, it's easy to see that along the time, higher is the concentration, lower is the motility and also the membrane integrity. So, it is very important to keep in mind, seminal plasma is not compatible when we are talking about extended semen shelf life. However, what about the sperm concentration itself? Is it important or not? The concentration that we're going to have in this sample that we want to keep for a longer time, is it important or not? The sperm concentration, it's relevant? Let's see this old study published many years ago. In this comparison, you have the motility along the 30 minutes, 3 hours, 6 hours, 12, 24 hours. And the concentration, 25 million per ml, 50, 100, or no dilution. Quite often, I'm asked about, well, if I'm gonna use the semen, I'll collect the semen right now and use it right away to the mare, it's important to add extender, or I can skip this part. I want to highlight this point. Look at that with me. No dilution. After 30 minutes, you already have a decrease in the motility. So after 24 hours, the higher motility you have in the lower concentration. So the take-home message that we have from this is concentration is important. As I said, to do the dilution based in volume is not accurate. And we have to try as best as we can to do the dilution properly based on the amount of sperm that you have per ml. Okay, I'm saying that it's important to add the extender, the milk-based extender, but it will act as a diluent in the seminal plasma. It's true. It will act as a diluent in the sperm concentration. Yes, it's true. But it does another thing as well to protect the sperm to the seminal plasma effect. Let's see it right now. There's a epididymal sperm that is there with low or no motility. Then you have these BSP proteins that are added to the ejaculation by the accessory glands, the seminal plasma. During this moment, this BSP will be attached to the sperm, removing many lipids from this membrane, increasing the motility, for sure, and they also remove some cholesterol. It is important, it's very important for the sperm metabolism, for the motility, it is. But it should occur quite quickly. And immediately after that, the sperm will be delivered in the uterus and will progress along the uterus until the fertilization site. However, when we are talking about the semen dose, they are together there. And if we don't remove the seminal plasma and if we don't add the extended milk-based extender, look what's going to happen. The sperm storage here, without this casein that is here in this situation, diluted semen and non-diluted semen. What we have in non-diluted semen, this BSP will keep removing, taking lipids and cholesterol from this membrane. And after a while, you'll be damaging that. In the other side, when you add the extender, these BSP membranes that do a good job, but they have to be removed or they have to stop. When you add a milk-based extender, what happens? It will be captured. Let's say the target will be the casein instead of the sperm membrane. So this is what I want to say. To add extender is important to stop or to reduce this effect. That is normal, but in the semen dose, it will be continued, and it's not good of this BSP effect that will be damaging the sperm cell. We have to avoid damage on the sperm, for sure. It's a very important, very specialized cell, but also very fragile. The metabolism, the sperm metabolism itself already produces oxidation, but also the environment, the osmolarity, and other things that has to be avoided. Because if we damage the sperm, what's going to happen? We reduce its fertility. It's something that we don't want to have. So many things can happen with these sperm. DNA fragmentation, many other things, apoptotic taints, decrease in the progesterone metabolism, and many other things. Most of them are related somehow to the ROS production, and most of them are related to this oxidative stress, the production of ROS in the normal sperm metabolism. So we don't want to have this sperm damage, and we have to consider if the oxidation, as I said, is the most important thing that occurring in the semen, we can somehow to think of antioxidation. So look with me The ROS produced here in the middle piece mainly, it produced lipid peroxidation and many other chains that can lead loss motility, loss fertility, loss of DNA integrity. So you have a general sperm damage due it. Based on that, some researchers thought, well, what about if I add antioxidants in the extender formula? Would it help to control this damage caused by this oxidation? Let's see. In this study was compared along 3 days the total motility of semen doses with antioxidants combination or without. And it's easy to see that the total motility of semen doses without control group, without antioxidants, it decreased along the time in comparison to the doses with antioxidants. That was quite high. So, what it tells us, the oxidation is part of the process. So the antioxidation, if we can control it, it will improve, it will reduce the sperm damage. All these things putting together, well, can result in the extended semen life. Another thing that we have been working since a while is this selection of the sperm through the centrifugation. And some stallions that are considered as bad freezers, they are not really bad freezers. Eventually, they just have a bad semen quality. So you have dead sperm, you have you have a low morphology quality. So if you remove all these bad things, these stallions can provide or can achieve a good semen motility after towing. This is what we have shown in this publication, showing that these stallions, these 6 stallions that were mentioned as bad freezers, this is the control group, their results using normal centrifugation, so really bad motility. When we selected them with single layer centrifugation, we got very good semen motility. What it means is that when you remove all this damage, those good sperm remaining in the sample can be easily manipulated, processed for freezing or for chilling, doesn't matter. So the main point, the main information that I want to provide, I want to share right now is removal of all these bad things is important to keep the sperm alive. What about the bacteria? It's another thing that we have to consider. Bacteria is not good. And to control the bacteria, we have to work in the hygiene, the good practices in the semen collection and manipulation, but also in the temperature and also in the addition of antibiotics. Let's see a comparison of all of these in this study. Here you have no addition of antibiotics, and the semen was stored at 5 degrees and at 15 degrees. When you compare both of them, you can see that 15 degrees, the progressive motility or the motility is lower. Why? No antibiotics and higher temperature allowed the bacterial growing along these 4 days of storage. 5 degrees were better, 5 degrees was better to control this bacterial growth. On the other hand, adding gentamicin, we could see that along 4 days, 5 degrees or 15 degrees were good to keep the sperm alive. So this is what we want to say. We have to play with. We know that dropping the temperature at 5 degrees can be harmful for some stallions. some sperm cells, but we cannot work only with 15 degrees without adding antibiotics. And we have to play with all of these together to be able to achieve a good result. So what about this bacterial remove? Is it possible? Yes, is it possible to do in 2 techniques at least, without adding antibiotics in this case, but removing the extra load of bacteria that you can have in the sample. In these 2 studies here, you have the amount of bacteria before and after single layer centrifugation. And you can see that's way lower after the centrifugation. In this other trial that we just produced that and was approved to be presented in ICAR in June 2026, it's using a new technique, microfluidic, that it consists, as I said, in the removal of. What about the removal of bacteria? Is it possible? Yes, is it possible? You can do it with or without centrifugation. But the removal of the extra load of bacteria is very important, because then you're going to have a more pure, in a higher quality semen dose to be storage. In these 2 studies here, you have this one using centrifugation. Before, this is a bacterial load before single layer centrifugation, and after in different bacteria. And in this one as well, using microfluid technique before and after using that. This second study was recently submitted to the ICAR meeting in Japan, and the abstract was approved, and we're gonna show it very soon. It was produced in Egypt, in Al Baida clinic with Dr. Omar Adel there, and it's a very new technique to remove the bacteria without centrifugation. That can be very useful and easier for the doctors in the field. Since this new technology became available, many different trials are being tested, trying to figure it out if it's useful, if it helps the fertility. And let's see one of them. This comparison is among 2 normal milk-based extenders and another one beyond. That is one new extender focused in long-term shelf life. So this is progressive motility, 17 degrees up to 7 days, 5 degrees up to 14 days. And what you can see here is that it's clear that Beyond has a higher progressive motility up to 7 days, 6 days at the end of the process. So it means that it's a longer shelf life. The same in 5 degrees group. You can see as well that Beyond has a higher motility. Also, in this study, they tried insemination and they got 61% of fertility. So it means that it works. Another study was tried with donkeys, also using this Beyond extender, and in that trial they added egg yolk. This is the trial using egg yolk, and also it worked at 5 degrees and at 17 degrees up to 7 days for donkeys. The big problem, or the big question mark, is it worth to be used considering the fertility? So does not make any sense to have this sperm alive or moving up to 14 days without pregnancies. And then I have one example right here that was published as well, comparing 2 stallions being used for insemination in different mares. Let's see. Storage occurred at 5 days, Stallion 1 and Stallion 2. These are at 5 degrees, sorry. This is 6 days, 9 days, 10, up to 14 days. Stallion 1 on day 6 had 2 mares inseminated. One became pregnant, another one did not. And along these days you have different mares being inseminated. For the Stallion 2, mare got in foal, both as well, and up to 13 days, one got in foal, the other one did not. So it were 14 inseminations, 14 mares. 12 of them became pregnant. 86% of pregnancy rate. So it works. It's a very good strategy to help us. And you can use different combinations. Selecting sperms as well, so you're going to start with a better sample. And you're going to dilute this sample with this advanced or long-term extender. We tried it last year, or 2 years ago better, and combining that motor device, this microfluidic technique, with beyond extender. And we also got good results in terms of motility. Let's see that. Was this presented at for a Association of Applied Andrology, Animal Andrology, 2 years ago. And this is the comparison. This is the control group, the motility of the control group. This is after vet motor selection, that in the group in 0 hours, immediately after the selection. So it means that motility is higher in the group selected and diluted with beyond. And at 5 degrees after 20 hours, the same. The group selected and diluted with beyond had a higher motility. So another thing as well, 2 or 3 years ago, say, we got this new technology in the Middle East. And with Dr. Maria, we decided, let's try to use it. Let's figure it out. We have one stallion that we could not keep the semen in storage for a long time. And we said, well, let's try with this one. Let's figure it out if it happens, if it can help us. Let's see. So this is the methodology that we used. This is the stallion that we were using. This is our control. So up to 1 day, 2 days, the semen's already gone. The motility was very low at day 3. But using the same stallion, but diluting with this new extender, we could inseminate one mare after 7 days. And the motility was 70%. And this mare got in foal. We could, with this trial, achieve the first pregnancy in the Middle East obtained with this new extender after 7 days of storage. And it generated this publication, or this abstract, submitted to one congress in Brazil in this case. However, for those still thinking that, well, it will keep the sperm alive, but it will generate only colts or only fillies, it's not true. Right now, we already have maybe more than that, but right now we have 6 colts and 5 fillies produced with this methodology. So it's not related with the gender, it's related with the sperm survival. Also, more recently, it generated one publication, the successful pregnancy using Stallion semen storage at 17 degrees, 6 days. So this is the publication. This is the very kind, very nice, cute foal that we got. So we are going to the end of this talk. The point is, after all the things that we discussed now, we can figure it out that now we have 3 kinds of semen dose. First one, the basic one that we just collect in the very hygienic way. Please do it. And then we dilute it normally using the concentration or using the volume. Okay? This is a very basic way to produce semen doses. But then you can also perform the centrifugation. The centrifugation can be done normally, just to remove the seminal plasma, or with some colloid centrifugation or other technique that will remove not only the seminal plasma, but the bacteria and the dead sperm, reduce the sperm with the not good morphology. Then you have two choices to resuspend the pellet with the normal extender, milk-based extender. So we're gonna have improved semen dose, or we can have the dilution done with the new extenders. This is one of them. Then it's not the basic semen dose anymore. It is not the improved one. It will be the modern semen dose that can reach up to 14 days. So this is the modern strategy that you can use at 5 degrees or at 17 degrees, and you have to figure it out. So with this in mind, it's time to think, okay, now I understand the tool, but how to use that? How it can be valuable for me in the daily practice? Well, quite easy. For example, let's think if you have a young stallion, 2 years and a half, 3 years, that you have to collect this little guy. And you collect this one on Monday, for example. And you don't have enough mares to use the roll ejaculate. Then fine, in general, you're gonna use part of it. And the remaining ejaculate, you're gonna throw it away. However, maybe on Thursday, for example, you have another mare. Then you have to collect this little guy again. So it will be a little bit boring, let's say, for him. Maybe it will not be good, you not collect the good semen. Many things can happen. But if you could, since last Monday, keep these doses in the fridge, it will be wonderful. They will be ready all the time to be used. This is just one example that came to my mind. I have other ones to show you. What about those stallions that are so aggressive? Do you want to collect them every day? What about those stallions that takes forever? The libido is low, or it's quite tough to collect them. You are wasting, for sure, many days of week, a lot of semen. Those stallions that you cannot collect because they are leaving the farm for competition, shows. Those stallions that are very, very far away and takes many hours, and you have to go there 2 or 3 days a week to collect semen for one mare only. And those stallions, this is a very interesting case. A friend of mine, for example, he has one stallion, an old stallion that does not produce enough semen for one dose. However, what he does, he collects the semen 3 days in the row and put all the semen together. And then he can achieve the minimum insemination dose. It's a strategy that it was helpful for him. Before that, it was impossible. Only if he decided to go to ICSI or, I don't know. But now he can use it with success. Also, what about this situation? When the semen already achieved your farm, but was delivered late, and you have the mare that is already ovulated. What about this situation? Maybe it's too late. The mare ovulated many hours ago. It's pointless to inseminate. But well, in this situation, you can resuspend, you can centrifuge the dose and resuspend it beyond and keep more days in the farm for another mare. Just an idea. Why not? Things like that and many others that up to you, you decide how to use that. So was the overview that I had to talk with you today, trying to explore all the aspects about how is it important, how does it work, and how it can be useful for you. So thank you so much, and it was my pleasure to talk with you.